Yeah, welcome everyone for the Marine Biology Night. I think most of you are probably familiar with me, but I'm Joan Lopez. I am the chair of the Marine Biology section for Nature Vancouver, and nice to see that some of you avoided being outside on this beautiful evening to instead be inside at your computer on Zoom for our presentation tonight. I'd also, first of all, like to acknowledge that we are coming to you from the traditional, ancestral, and unceded lands of the Tsleil-Waututh, Musqueam peoples, and that we are grateful for their stewardship of the land over the years. He is originally from New Hampshire, but he moved to Vancouver Island 8 years ago to study at the University of Victoria, where he earned his Bachelor of Science. And then he continued in pursuit of a PhD. He has been studying otoliths of fish species from throughout the Salish Sea, and recently also the gut contents of salmon for long-term predator diet studies. He will be talking about the otoliths with us tonight. And so please join me in welcoming.
Thank you very much. I will share my PowerPoint here and awkwardly try to get it into the presentation mode. So yes, hello, my name is Micah Quindazzi, and I'm here presenting otolith science of the 21st century, a bit of an overview of what I do, as well as just an overview of the field as a whole. And here is the backdrop. We can see many different otoliths from various species. I will get to a lot of these in the presentation, but if you want me to go back at any point to this slide and point out any of these that look really cool, feel free to ask. So about me, here I am on a boat at a 45-degree angle down a hill. I'm a PhD student in biology at UVic. My supervisors are Dr. Francis Juanes and Dr. Tanya Brown. I was not born an otolith. I started off as anyone does. I aged long-lived stickleback, and if you've ever seen them, they're these little tiny armored fish you can see swimming up the Fraser River along with the salmon. Here's one of their otoliths there. So one of my first projects dealing with otoliths was just aging them using these otoliths, which are actually about the size of a grain of sand, so quite difficult. Then I went on to, in between my bachelor's and my master's degree, I studied herring otolith morphology using a process that was kind of new to me, and so I got really interested in describing the shape and the form of otoliths by working on this project as a research assistant for a stint of time. And so a lot of my research now, so my research in my PhD really revolves around otoliths, specifically these two species, coho salmon and chinook salmon, are some of the otoliths that I study the most. And so I generally study the microchemistry, so the small scale chemistry across the otolith, as well as the morphology, so just the shape and form of the otoliths. And I do this mainly to track the migrations of salmon when they enter the marine environment and where they go to in the big black box that is the Pacific Ocean. And I also just do general otolith morphology for species and population identification across the Salish Sea. So getting to the point where you can pick up an otolith and tell which exact species of fish it's from. So background here, so otoliths. Oto just refers to ear, and lith refers to stone. And they're slightly analogous to our own ear bones. And so if you know, we have 3 sort of ear bones within our ears that aid in hearing sound that hits the eardrum. As well, in the inner ear of fish, they have these 3 otoliths that pick up sound in the water column and then transmit that into a sense that the fish can hear, basically. And what's really important here is they're not really bones, which is kind of interesting. They actually are, as the name implies, stones. they're mostly made up of a calcium carbonate. So basically like a snail shell or a clam shell that you might pick up on a beach, as well as other organic matter, such as proteins and some glycolipids. They actually form, they form in such a way that throughout time, they're forming over their entire life. And so you actually get like yearly rings. So, you know, on a tree, you can have these yearly rings that signify 1 year. And otoliths, you get the same sort of process where you can see here the dark bands in the otolith. Those are each year that the fish has been alive. This is a particularly weird one. This is a 5 to 6 year old coho, which doesn't happen very often. And we're not exactly sure what was going on with this one, but it was one that we found in our collection. So thought I'd share. And if you zoom in even further, you see that inside these bands, you get even tinier little rings. Each of those rings is a few micrometers, so 1,000th of a millimeter thick. And so what this is getting, what's happening here is the otolith every single day lays down a little bit of material. It becomes sealed away from the environment. So it's basically a time capsule of what it used to be. And then another layer is laid on top of it. And so effectively, you have a time capsule of the fish's environment every time it lays down one of these rings, which is quite cool. And so this is why they're more of a stone rather than a bone. And so, as I mentioned, otoliths are used by fish to detect sound. So they do this by using some tricks with density here. So seawater has a density of about 1.03 grams per centimeter cubed. Fish tissue has almost the same sort of density, just a tiny bit denser there. Fish bone even isn't that dense. It's only about 1.52 grams per centimeter cube. But the otoliths, they're almost 3 grams per centimeter cube. So they're almost 3 times as dense as the surrounding water. And so this basically makes the fish's body almost transparent to sound that's traveling around the water. And so when the fish's body will move, so the sound waves travel as pressure waves. If you've ever made like a splash or a ripple, sound kind of travels and propagates like that in the water column. The fish's body will move about the same amount as the water, but the otolith will lag behind slightly. And because there are sensory hair cells attached to the otolith, this lag can be detected by the fish, and then it can understand that to be a sound wave, and that's how sound detection with otoliths works within fish. And when you combine this with the lateral line, which you might have seen on a fish, it's a raised line that runs down the middle of the body. When you combine these two systems, it's called the octavo-lateralis system. And it's basically just the system that's responsible for sound perception within fish. So some background of otolith science. It's a fairly old field, and I've put 2 dates up here of 1881 and 2021. So 2021 being today, but 1881 was really the first time anyone described what the ears of fish kind of looked like, the interior ears. Some German fellow named Ritzius. And so some other important dates, in 1899, that's when you get that first description of those yearly rings I was talking about before. 1914, a fisheries scientist actually started using those years to help understand the stock of herring in the North Sea to figure out why some years were really good and some years were really bad. So that was using that description of otolith annuli in order to uncover that. In 1929, they further validated those as occurring yearly. In the 40s, we start seeing otolith studies that involve measuring the otoliths in relation to the fish size, as well as the fish's age and things like that. In 1960, they discovered kind of the exact composition of the otoliths. So they're the form of calcium carbonate, unlike a mollusk, which is calcite, which is a really less dense. calcium carbonate. They're made of a calcium carbonate form called aragonite, which is the most dense form. Anyways, in 1971, the first description of daily rings came about. 1989, the first otolith microchemistry paper was published. And then in modern times, 3D otolith morphometric studies were published, as well as detecting marine migrations using microchemistry. So these two last things that are kind of more recent things on this axis are things that I study, but it's built upon a very important foundation of prior research. And so a lot of papers, there's a long history there of aging these fish. So here's that stickleback, and I've aged it here. So those black lines indicate years. So this stickleback was about 4 years old. A lot of papers deal with aging fish. A lot of papers deal with determining stocks of fish based on the shape of the otolith. And then more recently, some studies used laser systems like the one pictured here to study the small scale chemistry across the otolith. And so the outline of this talk, I really want to discuss the two fields of research I partake in within otolith science, those being otolith morphometrics and otolith microchemistry. I will describe how it's done, what new innovations exist and what is being developed, as well as the most important one here, why should anyone care? And my sort of thesis statement here is otoliths provide a valuable window into the lives of fishes that can help us to better conserve them and to assess the stressors that impact them the most. And so now I'll get into otolith morphometrics here. And so morphometry refers to a quantitative measurement of forms, and a form just means a size or a shape. And this could be as simple as measuring a fish or just measuring the length and width of an otolith. This is what is meant by morphometrics, it's just these measurements. Morphology is related, but not necessarily the same thing as morphometry. It's just the study of form, and it doesn't have to include measurements in it. And so things that scientists have known for a while is we can really easily measure otolith length, their width, the perimeter, and the weights. And so you can distinguish species based on the overall morphology here. As you can see, for example, a coho differs a bit from a chinook in terms of how the otolith looks under a microscope. So this is kind of what a lot of scientists call the first wave of morphometrics was doing. It was just about distinguishing species from each other and doing simple linear relationships like the length to width of species. The second wave of morphometrics was more like this. I'll describe this here, but essentially it's a truly multivariate study. So we're not just considering a length to a width per se, we're considering length to the width, to the perimeter, to the weight, and things like that. And so that what this image here shows is basically, I wanted to characterize the when you have two objects and they're not symmetrical, they're just known as asymmetric. And so this, I wanted to describe how a lot of different asymmetries and a lot of different types of measurements differed between fish that had an abnormal otolith versus like normal otoliths. And what happens here is when you plot all of these variables onto a two-dimensional plane, what matters most is where these colors here separate out, and you can see the yellow and the orange almost completely separate apart from each other, indicating that the abnormal shapes are differing than the normal otoliths there. So that's all this is really just showing here. And then from the second wave there, the third wave that people call morphometrics dealt with dot drawings, they call them. And so you can kind of see here, this stickleback's body, there are a bunch of different dots drawn on it. And so what this allowed people to do in morphometrics is to determine how each individual dot on an outline changes between populations. And so you can characterize the different forms between two populations based on some like significant features. And so it allows us to characterize more of the form than we otherwise would have done. The problem with otoliths, in particular with this method, is trying to draw dots on these otoliths is a little tricky. Like we could maybe draw one here, another here, and two here, maybe one here. It's not really helpful. It's not something we can really do with otoliths, which is why there aren't a lot of studies that kind of try to do this. It does lead to the final wave of morphometrics, which is just doing the full outline of an otolith. So these are just different rockfish species. It's a little tricky to describe exactly what's going on here, but basically imagine if I drew this herring otolith, you see this red outline around it. And if I snipped it in one place and tried to lay it on a flat line and then compared those flat lines to each other, this is essentially what those graphs are trying to show, is they're taking a perimeter and turning it into an XY plot that we can compare against other plots. And so that's all that's trying to do. Other things that people have been trying to do, they're trying to automate things. So automation is taking over even in science, and then doing 3D sorts of studies. And that brings me to an important point. All of the photos I've shown you about otoliths have failed to get across one important detail. So we have two rockfish otoliths here. They're not flat. So they're actually kind of like bowls a lot of the time, especially really large otoliths. They're definitely bowl shaped. They have what's called a sort of information in the Z axis. What that means is I can't take a photo of an otolith that captures all of the ways that it can differ between different populations because it's two-dimensional. So we need that third Z dimension in order to fully capture what's different about them. And so to do this, we use what's called a micro CT scanner, pictured here. This thing can fit quite reasonably on your desk. And the best way to describe it is you've seen a CT scanner before. And so now imagine instead of a person, I've put an otolith in there. And so that's basically what this kind of method is doing here. And so this is kind of what I've been doing. One of my most recent papers that came out in the Canadian Journal of Fisheries and Aquatic Sciences, I had in panel A here, a 3D printed mold that I stuck coho otoliths in. And then you can see a CT scanned image of it in panel B. And then I can separate those otoliths out into individual otoliths, which you can see in panel C. And then panel D here shows what an individual otolith looks like when it's three-dimensionally scanned. And so that's the kind of stuff that I'm trying to do. And so this is useful. This kind of method's useful because not only is it three-dimensional, but this was automated. The guy that I went and did this with, we went and ate a sandwich, and when we came back, all the data was collected for us, which, you know, that's amazing. So other advancements in this field. So a lot of care and attention has been put into getting the most we can out of 3 dimensions. So what I've kind of described with the otoliths that I had before was kind of dealing with just volume mostly. I haven't been able to capture the full extent of the shape. And so that's what a lot of new research is getting into, is just more actively including the Z axis into prior existing methods, as well as machine learning. Furthering automation is helpful because it allows us to, as scientists, to do other tasks instead of just clicking and measuring otoliths all day. And so it also helps with species identification, because if I can scan an otolith and a computer can tell me with really good certainty what it is, it means that we can incorporate this technology into fisheries management to help determine if what species are being listed as when they're caught are actually what they are based on what the computer is saying. And so that's quite useful. I will pause here and just say for where I collect otoliths, I kind of collect them everywhere. Mostly I collect a lot of salmon otoliths directly from hatcheries or from what they call test fisheries. So right at the mouth of the Fraser, there's a test fishery that happens. And so that's where I get a lot of my Fraser River salmon from. And then other samples I get a lot from just working with people over at iOS, and then anything that comes up in research trawls that they think I want. I've kind of become this resident otolith person that people just hand me samples, which is kind of an interesting gig to have as a younger scientist. So anyways, so the so what here? Why should anyone care about what I've kind of described? So this field's really useful for, as I was saying, identifying species and populations within these species. In a fisheries context, it can really help us see what sort of important stock structures there are going on. So it's not like there's just one species of fish and that species of fish all does the same thing. There are different sort of groups, and sometimes those groups within the broader species are more important than otherwise we would expect. So for example, one part of a population might be commonly consumed by an enigmatic predator. So we know, for example, killer whales will select certain populations of chinook, mostly because those populations are larger at size. And so they want to get the most energetically cost-efficient meal they can. And so they will target certain stocks of salmon. And it's generally cheaper than a genetic analysis, especially with the good success rate they have. So if you're going to get the same results by running a $40 test that you could with a 40-cent test, you're going to pick the 40-cent test at scale. Fluctuations. So basically, this one's a little more sort of difficult to grasp in a certain respect, but basically, the more asymmetric something is, the more likely it has something stressing it in the environment. And so if we note higher amounts of the asymmetry, it means the stressors are higher as a result. And so, yeah, the more varied the asymmetry, the more likely there's a stressor in the environment. And so that's that for morphometrics. And so I'll move on here to microchemistry. And so what is microchemistry? It's really just referring to sort of small scale chemical concentration differences that we can detect at like a small size. And so typically I measure things on the scale of 30 to 100 micrometers. So, 3 to 10% of a millimeter. So at a very small scale here. And so how this is measured, we use the method's called laser ablation inductively coupled plasma mass spectrometry, or LA-ICP-MS. And so a good description of it, imagine we took an otolith here and we have this box. So there's this tube connected to a box, and there are two rods in this box and they're charged. And so basically those rods will only allow a specific element to pass through to that blue oval, which is the collector. And anything that hits the collector is read as an element that has been counted. So we take a laser, we fire the laser across the cross section of this otolith. It leaves this nice burn mark where it passed through. And so all the elements that are trapped with all those calcium carbonate layers, they get released. And so this orange element, it doesn't have the right mass or the right charge. It doesn't get through the two bars there. The purple element also does not go to the collector, but this blue element flies down and gets read by the collector. And so we would get a count of 1 of that blue element. And so this is reading elements specifically here. So things like oxygen, nitrogen, carbon, things like that. And so here's what it actually looks like in process. The glowing sun looking thing in the screen is the laser that's slowly shooting across the otolith. If you look a little bit more on the left-hand side of the screen, you can see dark and white bands. Those are those daily rings I was talking about, that alternating layers being laid down every single day. And so within those layers is where all of those elements are. And so what's useful about where fish are, different parts of the environment will have different characteristics, which means they have different collections of elements in them. So things like salinity varies quite distinctly between the Strait of Georgia and the west coast of the island and the west coast of Washington. Temperature changes quite drastically within certain regions, especially within, Juan de Fuca Strait versus the coast of Washington. We have right at Vancouver Island, we actually sometimes get the California current and sometimes we get the Alaska current, and it changes throughout the year. And so that will change what elements are present in the water. And then one that's also fairly well known, here's a map of iron concentrations in the water. And so right close to shore there, near Kodiak and stuff like that, we get slightly higher iron levels. And then as soon as we get out into the Gulf of Alaska, there's almost no iron whatsoever. Because that's like a very specific element that kind of differs between the sort of two regions here. And so if we looked at all of the elements at once, as you can see, basically the only thing that matters here is the orange group and the blue group separate out. And that allows us to distinguish between the two by looking at the elements within their otoliths. And so that's kind of a summary of what's trying to be done with the method. So a lot of the first uses here were to distinguish between sea run and freshwater resident trout. So if you had like a juvenile rainbow trout, sorry, let me back up one second here. There is a distinct difference between what seawater looks like to the chemicals within it and what the freshwater versions look like. And so you can kind of see on the top graph there, you have black, which are all the freshwater progeny, and then you have the lighter gray, which are all sea farm. And so basically you can see they separate out there. But it goes one step further. We can actually take a juvenile, like this juvenile rainbow trout, and based on the very core of the otolith, we can tell whether its mother was a rainbow trout or a steelhead. And so which one was its mother? Well, we can tell right at the very core of the otolith, because the steelhead will develop the eggs out at sea in seawater, and the rainbow will develop its eggs in the freshwater. And in some populations, those two mix. And so you can actually see some interesting crossover during those sorts of time periods and in those sorts of populations. And so interesting work is being done with BCCF, with Jameson Atkinson, and they've done some interesting work with these sorts of questions. But this is probably the longest standing use of otolith microchemistry, and this is a figure actually from one of the first papers that ever came out on this subject. And so some other interesting things that they've been able to do with this is, for example, trying to figure out the benefit of migrating to sea versus the benefit of staying in the freshwater. And so we can see here, this is based on Dolly Varden. And so you see here where in the graph that's available here, we can see a 0 and a 1. And so the 0 represents fish that have remained in the freshwater, and a 1 means they've gone to sea once. And the y-axis here is just the size of the fish. And so we can see is if you've gone to sea once, you're a lot larger than your other counterparts that never went to sea. And if we go further on, so one other year, you still see migrating out to sea has this size benefit that the residents don't have. But once we get to a certain age, everything balances out. And you can see at these older ages, if you had never gone to sea, you're actually still about the same size as your contemporaries that did go out to sea. And so there's a bit of a cost benefit analysis here, kind of in the fish's mind. They're weighing the odds of being picked off by a predator trying to migrate out to sea versus not being as large as those that did go out to sea. So they're kind of doing this cost benefit analysis. I can give an example of like what a cost benefit analysis might look like using coho, because this is something I understand quite a lot better. So imagine we have a coho juvenile here. And so maybe it had a lower relative growth during its first summer at sea. And so it's quite small once it gets to September. And so it's got to figure out, do I remain within the Strait of Georgia? And I could die quite easily. There's a lot of size-based mortality events that can go on. Or do I risk it, migrate and try to pass all those sea lions out the Johnston Strait? And if you make it, you have higher growth just at the risk of higher predation. So you'll be a larger fish when you return, and larger fish produce more young. Or if you grew a lot during your first summer at sea, maybe you're a bit bigger, you can elect to remain. You have a lower growth, lower predation risk though, and you're smaller size at age and you have a lower reproductive output. And then there's also this third option. It's males only. I'm sorry, everyone else, but they're fast freshwater growth. And so sometimes they just return to the freshwater soon after they finish their first summer at sea. Those are jacks. And so they kind of fit on this nice little thing. But basically, a migration choice here is related to survival and reproductive output. And what's important is we know this by looking at those otoliths and looking at the microchemistry. So this is something that we can determine with anadromous fish, so fish that migrate out to sea and then return. But what about fish that are fully within the freshwater or fully within the marine water? Can we detect these sorts of migrations? For fully freshwater, we can. And this is because across BC, if you actually look at a map of the rock deposits that the rivers run on, all of those bedrock deposits have certain elemental signatures. And so those vary at really large scales in watersheds and also vary at really small scales. So just specific rivers within watersheds. And so imagine here we have the Fraser River highlighted in red. And we catch a coho here that's right at the very mouth here. We've caught it, just off the wharf in Vancouver. And we want to know where it came from in this very large watershed. So we take its otolith and we have this little graph here is going to represent some elements in a second here, but basically we take the otolith, we start shooting a laser, and we see this little signature, this red signature that corresponds to what we know the Fraser River should look like on an otolith. And that's not surprising because, we caught it right at the mouth of the Fraser River and it looks like it's been in the Fraser River. That's not at all too surprising. Now we look at all these tributaries that we know the signal of because we want to know which of these tributaries it came from. And so we ablate the otolith a little bit more. We get a little bit more of the graph and okay, we start seeing this blue signal here. Okay, and we ablate a little bit more. And now we've reached the very center of the otolith. This is where it was when it was just born. And we see this light blue element. And so if you kind of look at the river system, and I apologize, this looks way smaller than I thought it would. You can kind of see that goes towards the Thompson River system. And so we can see that our fish actually originated way up here. And so that's something we can tell by just looking at the elements within the otolith. And so that's fairly interesting. And this has been done a lot in BC. So this has been done for the Fraser River system and coho by Mark Shrimpton before in the past. So that's pretty interesting. And so we can kind of consider the life history here of a salmon because we're kind of curious now, what about the marine stage? Because that's fairly interesting. We get the egg, they go to a fry, they become a smolt, they leave, and then they kind of become this subadult stage before they finally return as an adult. And that subadult stage could be the longest part of their life history for a lot of these species, especially chinook. They can remain years out at sea. And so can we detect where they go? You know, we have these migration maps of where fish kind of go in the ocean, but it's really hard to kind of tell where they've gone just by any fish. I'll get to how this map itself was constructed, so you can kind of see the difficulties with these migration maps. More recently, it's been found that if we look at a lot of elements all at once, we can really detect marine environments between various species, so we can see where they've moved in the marine environment. This is only something that's happened fairly recently. It's been done in anchovies, Atlantic herring, and actually in swordfish as well, and I think tuna maybe, if I remember correctly. So that's quite interesting that they've been able to do it for these like fairly well-known marine species. And so my research specifically uses this kind of approach to track Pacific sand lance movements and then coho and chinook migrations. This slide is just kind of getting across that one caveat I have to put in here is why I need to collect some extra samples out at sea. And that's just because the elements in the seawater have to pass through 3 sort of barriers before they're absorbed into the otolith. And so this is a bit more technical than I was hoping for a slide presentation thing here, but this is kind of all I had. Basically, all to describe it, it goes from the marine environment into the blood, into the liquid around the otolith, and then into the otolith. So it's not like a straight one-to-one representation of what the marine environment looks like. And that's why we use a lot of baseline samples. So samples that were caught in the region where we're trying to get some signals from. And so, yeah, we need to get baselines for different regions to know what the chemistry looks like in the otoliths if a fish was in that region. But fish move a lot, sometimes a whole lot. Chinook can move, I think it's like 100 kilometers in a day or something ridiculous. And regions can also change with different seasons, so that's not helpful either. Luckily, though, some move a lot less than others, and sand lance barely move at all. As adults, they kind of stick to a very, very small range of habitat, mostly because they're almost trapped on islands. They don't have a swim bladder, so it actually costs a lot of energy for them to swim very far, and they're also very small fish. So they tend to stay around their neck of the woods throughout their entire lives, meaning their otoliths are really useful for these sorts of baseline samples. Anyways, I thought I'd mention sand lance work because it's quite interesting, but really salmon pay the bills for me. And another important thing about them as well is their abundances within the Salish Sea have been declining quite significantly since the 1990s. And that's because we really love eating them, as well as a lot of marine, and the marine environment has become a lot more hospitable for juvenile chinook. And it's commonly ascribed that marine survival has decreased for the smallest stages of these fish. And so it could be the case that it's harder for them to survive to adulthood without even our input. And so one important thing to know, though, is another possibility is that they've started to migrate in different locations as well. And perhaps those migration routes have a large amount of predation associated with them. So it's something that we need to figure out in order to be able to tell how much the marine environment has decreased in quality for these fish. So typically the way this was done in the past, back in the 1960s actually, to track salmon migrations, they would put this thing called a Peterson tag through the back of the fish right under its dorsal fin. And actually, if you ever seen these marine migration maps, that's how a lot of them were constructed, because before 2019, the last high seas cruise that was capturing salmon off the coast happened back in the 60s. So it's been quite a long period of time. And this was the height of technology they had to track these migrations. And so in order to tell where a salmon migrated to, you caught it on a boat in the middle of the Pacific Ocean, you put a tag in it, and then you hoped that someone would find it in the river and report its tag back to the Department of Fisheries and Oceans or Alaska Department of Fish and Wildlife or whoever else. This didn't really have a very high success rate, but it was successful enough that we kind of generally understand where they moved in the marine environment. More recently, and by recent, I mean the 1970s, coded wire tags were put into a lot of salmon. And so these are just little tiny strips of metal that have a number written on them that usually gives you some information like where it was reared at, which hatchery it came from, as well as when it was tagged, when it was released, information like that. And you're supposed to return the head of any adipose clipped fish that you catch, because those are the ones that are going to have these tags. And then we kind of have two data points. We know where it came from and we know where it was caught. And so we can kind of create some migration map information as a result. Here we can kind of see the numbers that are kind of coming down the sides, and then the colors all represent like a river system, and then what percentage of that population migrated to something like the Strait of Georgia, which is that yellow color, or Puget Sound, which is that red color, or this west coast of Vancouver Island, which is that green and purple color. And so that's kind of what we've been able to glean from those coded wire tag migration information. But one thing that was fairly kind of interesting is we still need this physical interaction with the salmon twice. We need to tag it, and then it needs to be caught again. But what if we could just take any salmon that we come across and be able to tell where it's from? What if there was an internal tag the salmon already had? And so we can kind of imagine, for example, you have two coho. One goes off to the west coast of the island, one stays in the Strait of Georgia. And then again, that migrant returns. When it's back in the river, how are we supposed to know where it was from originally? And we can reimagine the otoliths here. And as a fish kind of goes through its life, all of those layers of otolith are being added onto each other, and those elements are incorporated into the otolith. And this keeps happening throughout a fish's life. And so we can imagine then, back to this example, their otoliths will have different microchemicals within them, different element concentrations that are a record of where they've been. And so they become our internal tag. And so any fish with an otolith, which is basically all of them for salmon's sake, will have a record of where it's been. And so we could use this method on any species of salmon, whether or not we've ever interacted with it before or not. And so that's kind of the best pitch I could give for sort of microchemistry here and otoliths. And so this kind of gets into my project. A lot of what I do is I'm trying to determine differences in otolith trace elements between salmon from different marine regions. So those are my baseline salmon samples from the marine regions. And then as well as I look at the marine migration in returning adults. So I just compare those returning adults to what I know the baselines look like, so I can determine where those adults were when they were migrating in the marine environment. And so this work is fairly extensive because chinook especially migrate very far. And so these are the kind of regions I have covered here. A lot of my work is with DFO in BC, do a lot of work with the International Year of the Salmon for the offshore fishing zones, which they have been implementing and they're going to do this year and the next couple of years. I work with Washington Department of Fish and Game down in Washington to get samples from Puget Sound and the Washington coast. And then I have friends up in the Alaska Department of Fish and Game to get samples from the Alaskan coast, from Kodiak down to Juneau. And so that's kind of a summary of what I sample out in the marine environment for my baseline samples. And when I compare the baseline samples, so far, so good. I was able to, what I've run thus far, I've been able to tell the difference between coho that reside on the west coast of Vancouver Island and the Strait of Georgia. And that's kind of what this figure just shows. The Strait of Georgia is here in the pinkish red versus the west coast here. And you can see those two element signals are so vastly different. There's almost no way that they overlap, which is quite reassuring. It means that we could probably quite easily tell the difference between different migration regions in returning adults as well. And so I've been also collecting a lot of spawning coho from various hatcheries, and now I'm collecting chinook as well since I rolled up to a PhD back in May. And so future work is happening on both of these species at most of the hatcheries in the future. And this work's all being done with BCCF, PSF, so the Pacific Salmon Foundation, and DFO. I'm starting with a lot of the coded wire tag populations to compare overall results between what we can tell from the coded wire tags and what we can tell from the otolith chemistry. And so this is just kind of a map of the different regions that I am going to for coho and chinook. So there's quite an extensive amount of it. So I do come to the mainland from time to time to come visit some of the hatcheries out there. Yeah, and the best part about this work is the samples come to you, which is another thing that scientists really love, is when you don't have to put a lot of effort into getting your samples. And so like these are kind of nice little maps that I also have in here. This is just kind of what we expect fish to migrate to. So this is with coho. I could provide these maps to anyone after the fact because I don't think I have too much time left. So, but yeah, if anyone wants these maps, I'm happy to share them. Yeah, again here though, so what kind of comes up time and time again, especially in my funding proposals. Otolith microchemistry really can help improve our understanding about where fish are and how they are moving within their environments. We can also look at related things such as growth rate and genetics to understand why they're moving the way they do. It's helpful for determining the paths of fishes as well. We can use this sort of knowledge to keep blockades clear of where fish are moving. And so what's actually interesting is I know for a fact that hydroelectric operators are considering and doing this kind of work to help determine the least detrimental place to put dams to not block the migrations of fishes. So that's kind of interesting. And so this can help us to really figure out how much of a population migrates to different regions and which of those migrations is the most successful. It can also help us to know where we need to stop fishing in the marine environment to stop overfishing certain populations. So that's something that wasn't really, we weren't able to do that really before. And then we can make really informed comments about actual marine survival rates of the juveniles to the adult life stages. So conclusions, I hope I've just not bored you all to death. No, seriously though, otoliths really have a long history of use for marine and fisheries ecologists. Recent advancements in otolith morphometrics and microchemistry really can help us better to understand fish. It can really help us to deal with species and population determination. We can look at migrations of fish at various scales and regions. And with that, are there any questions? Because that's the end of my presentation.
I had a question early on about you had a graph where you drew a line around the perimeter and but I couldn't figure out what the y-axis was on that graph. Right where the x-axis. I think so.
So just like with the herring there?
Yeah. So you drew some. You pointed out that you're making graphs where you're going around the perimeter of the otolith to characterize them, but I couldn't figure out what it was that you were measuring along the perimeter.
Oh, yes. It's really hard to see on this figure because, but basically the box here, the longest axis is measuring that length of the otolith and the width as well. The perimeter that it's drawn on there is just measuring actually just the perimeter on this one. But if I wanted to turn it into a two-dimensional sort of axis, what you can do is you can cut the line in a certain location and then kind of bend it out into an axis. And what that is kind of showing you is what the relative turn is that you're following. Like if you're following along the line, how much you have to turn to stay on the outline is kind of what those two-dimensional forms of those look like.
Oh.
That's kind of the best way I think I could describe that.
Thanks.
Yes, hormones will control the deposition. So if like a stress event can actually prevent daily rings from forming. And then as for what triggers them, it seems like it's related to growth in some sort of fashion. It does seem to be circadian because you will see fish undergo these sort of circadian patterns. But in some species, they don't actually form these daily rings at all. And it's not quite clear why a lot of the time, because it's not like deep sea species where you wouldn't expect a circadian rhythm. It's almost weird species like herring don't do it, which is quite weird.
There was a question there too about how big are they, which I suppose would vary with fish, but in general, and do they show up in sediments?
Those are both great questions. So otoliths can range in size from the stickleback. Actually, you know what? I'll go here to the title. I'll tell you how big each of these are. So most of these, so the top two otoliths in the upper left-hand side, those are both actually from myctophids, so lanternfish. Those are about 3 to 4 millimeters long. The coho otolith, which is to the right of that myctophid otolith, is about 6 millimeters long. The one that's in the upper right-hand corner there is from what's called a barracuda, which it's hard to describe what those are. They're like little mini barracudas. They almost look like a sand lance and a barracuda mixed. That's how I describe them. And so that's about 4 millimeters long. Sand lance otoliths are about 1 to 3 millimeters long, and that's in the bottom left. Next to it is a deep sea smelt. It's about the same size. Rockfish otoliths can be quite huge, actually. Some of the biggest otoliths I have are rockfish otoliths, and they're about 2 centimeters long. Chinook otoliths are about that size as well. The largest ones can be 25 millimeters long. The bottom right otolith is from what's called a king of the salmon, which is a giant ribbon fish. If you've ever seen them, they're quite stunning. They're metallic. They almost look like a metallic noodle with giant eyes and a gigantic red sail. But they also have the smallest otoliths because those are only 2 millimeters long for this absolutely monstrous fish. Oh, I think I missed like one right at the very beginning. Do people do studies of talking to fish? Fish, yeah, so fish do communicate with each other. Salmon almost, what it would sound like to you if you could hear it, it almost sounds like they're burping at each other. And they can quite clearly hear each other, and they can determine what those sounds sort of mean. And people think that they communicate through noise in this way to, like when they're schooling up and staging. If you've ever seen salmon kind of coming into the river, it's really well organized. They do it really quickly, all at the same time. And it's probably a portion of it is sound, and then another portion of it's going to be visual. But I think sound really is important right when they're still way further out at sea and they can't see each other nearly as well. And so there is a lot of research that's going into that, and members of the Juanes lab do study fish sounds quite extensively. And so there's a website for that as well, which escapes me presently, but it would just be under the, if you looked up like Juanes lab fish sounds, you could probably find the website that's related to it.
I have a completely different question. So have you heard the story recently, was on Quarks and Quarks, I think, about woolly mammoths and microchemistry of tusks and to track their travel?
I have not heard that, but that makes sense to me. They've done the same sort of thing on historic fish. They've done they've been able to track migrations of ancient fish, which is kind of interesting. But yeah, woolly mammoths, that totally makes sense to me. It's a really interesting method because there are a lot of structures that are really well preserved in terms of keeping the elements of where they've been. So makes that sort of methodology useful. And then for sediment deposits, yes, you see them a lot of the time. So if you've heard of like shellmen before, you kind of get like that black soil with clams that were discarded by First Nations a long time ago. Inside those piles, we actually have recently been finding a lot of salmon otoliths from old fish that were caught, eaten, and their carcass discarded. We have those otoliths from those fish that have survived. And so a lot of work is being done with the UVic archaeology department out in Banfield. And they've collected a lot of otoliths, and I'm helping them to try to identify which species they came from, because we're talking 5,000 to 6,000 year old otoliths. So they're a little crumbly by that point, but they're still there. And you can do the same thing from like bear poop can have fish otoliths in it sometimes. Same with wolf scat, orca scat. There's actually an entire catalog of otoliths from sea lion scat that was developed by NOAA. So it's definitely something you can do. They survive probably the best. I'm actually pretty sure the sand lance I have on the on this slide was actually in the stomach of a salmon, though it was still intact by the time we pulled the otolith out. But we get I've dug through salmon intestines, as I was previously pointed out. I do study some salmon diets, and even in the intestines, when no other part of the fish is really remaining besides the bones, we see the otoliths too. So those survive fairly well.
There was
Oh, sorry, go on.
oh, I was just going to that there was a question, but you probably just thought about the fish arriving to the hatchery.
Yes. So fish arriving to the hatchery. Do you look at lost fish? We do take the genetics of a lot of these fish as well when they're coming in, so we can verify that they actually did come from the hatchery. And so that's something that we do keep track of. It's not often we see a lot of straying. We only see it in kind of particular systems. So some systems around Cowichan can get some Cowichan hatchery chinook will come back in there. So it's something to be wary of. But what's actually really funny is if you go to a lot of these rivers, you can actually pretty easily tell a interloper kind of in the system. You can pick out and go like, you look really weird. You don't probably belong here. Fish in like Campbell River, for example, all of those chinook are really yellow, which is very strange. But if you saw a chinook that didn't look anything like that, it's likely that that could be a straying fish. It's hard to describe this. It's something that if when you see it, you can understand it a little bit more than how I've tried to describe it here. But I hope that I was able to describe that fairly well. How much does it cost to get an otolith out of a head? As much as I'm able to freely do it myself. So I have a good working relationship with a lot of these hatcheries, and so I can go and sample otoliths. The most it costs is basically the amount of otoliths I collect divided by the gas that I spent to get out there. So fairly cheap.
And how do you go about I mean, maybe without getting too gory into it, how difficult is it to get the otolith out of the head?
That's a good question. For salmon, it's actually really easy. I can almost do a method that's they call it a haircut method because it's just a little bit off the top. You just take a little bit off the top of the chinook's head and it quite easily exposes where the otoliths are. They sit like right behind, behind where the brain is, basically underneath. So as soon as you expose that, you can pick them out pretty easily.
Oh, yeah, that was somebody else had asked that. Could you please point to the location of otoliths on one of your pictures? Are they floating in a fluid-filled cavity or anchored by fascia to another structure?
That's a good question. I think I have at least a picture here of what this is. So on the right there, this is called a semicircular canal. And so these are fluid-filled sacs that the otoliths kind of float around in. And so each of these, if I were to point out where it is on a fish's head, oh, do I even have a fish in this presentation? It's one of those things, I didn't even think about it. Oh, here we go. At least I have an ulican here. So I can just slideshow, play from current. Can you see my mouse here?
Yeah.
So they would sit relatively right behind the eyes and before the, this is called the operculum. So this is where the gills are. So right about here on a fish's head is where it is. So just behind the eyes, right before the operculum.
And there's two in each fish?
So there are two sagittal otoliths, which are those big ones, but technically there are 6. Just 4 of them are really, really tiny and no one uses them, so we don't talk about them.
Do cartilaginous fishes have them as well?
So that's a great question. And this is another thing that's kind of come along with how otolith science has developed. A lot of older scientists, like back in before the 1960s, used to call otoliths statoliths. And so all fish have statoliths, but they tried to distinguish otoliths from statoliths because they're so much bigger. Within a shark, they will have a what is called a statolith, but they're so tiny, no one really uses them for any purpose, and they don't kind of grow in the same way. They're not really the same sort of undergo the same sort of processes, and they're not really used in the same ways. So with sharks, it's more like what a statolith is kind of used for is to just make sure that it's upright, basically. Its orientation with gravity is kind of what it's used for, but otoliths are used for more than that. So that's why there's a bit of a distinction now.
All right, I don't currently see any other questions in the chat. You are welcome to unmute yourself as well if you like, if you just wanted to ask Micah a question. I had one for you. So when you're looking at the microchemistry and you said, well, a certain element only will get through to the plate, does that mean that you have to run multiple tests and adjust it for each element that you're looking for?
So that's a great question. What's nice about it's hard to get through it all, but the laser system, it essentially switches elements that it's reading every 1,000th of a second, essentially. And so it's all happening really fast. So that laser's shooting a lot of material up the pipeline, and the lasers, the rods that are controlling what elements can go through flips and changes. And so basically it comes out to like every 1/1000th of a second, we can get a reading of an element or so.
Wow. Well, that's isn't technology great? Right? There was a time you would have had to change it, what you're looking for.
Yeah, there
ontogeny or-
are some systems where they're so precise and they still use that kind of method. And so they do go and they have to change it for every single element. And I do not envy people that do that.
Anybody else with questions? So I know it wasn't part of this talk, but I was kind of fascinated by the idea of the, the gut studies that you were doing as well. And it sounds like say, not to totally change the topic, but it sounds like you were just getting this from even like sport fishing guides and so on, getting the stomachs.
Yeah, so the way it was working out essentially was my colleague who started the project used to actually be a fishing guide out of Campbell River before he was doing his PhD. And so he built a few connections with some friends. And so he was like a lot of anglers love to know what their fish are eating, mostly out of interest of what fish are there, but also to kind of get the edge and see what salmon are going after the most or what's most available in the water column. And so he took that knowledge and started asking people for stomachs from recreational buddies he knew, as well as he made new connections across Vancouver Island to places through things like fishing derbies. And so this built up into a project now where we've sampled over 3,000 stomachs as of a couple days ago. We're just at over 3,000 now.
Wow.
And so, yeah, it's built up into this very large thing. I've sampled through 600 to 800 myself. I don't have an exact estimate on the numbers because, time flies away from you at a certain point. So.
Wow, that's also sounds like a really, really interesting study. Because yeah, you're right. The sports fishermen, especially, they want to know what will give them the advantage to catch the next fish, right? Yeah. I don't see any other questions. I'd like to thank you so much, Micah, that was a really, really interesting talk. It's very tiny and detailed structures, but fascinating what you can learn from studying them. And I think that everybody learned a lot about that tonight. And I know that there were a number of people that had sent me an email going, I hope the talk is going to be recorded because I'm not at home. So thank you also for agreeing to recording it. I think you're going to have a bit of a follow-up audience after the fact as well.
Sounds good. Yeah, thanks for having me. It was definitely, I've been always interested in talking to more people about this.